Vale, pues empezamos, ¿eh? Lo lanzo. Good afternoon, and thank you all to all of you for attending the webinar today. Today we have the webinar from the Regulation of Gene Expression and Genome Dynamics, led by María Gómez Vicentefranqueira. Thank you, María, for organizing this webinar, and please introduce you today's speaker.
Okay, thank you, Mar, and good afternoon, and thanks all of you for joining us today, and especially thank you for Félix Machín, which is today's speaker, and he's connecting us from Tenerife, which is very nice. One of the very nice things that the webinars allow us to do. So Félix did his PhD at the University of La Laguna in Tenerife. And he also did his, he did his studies there, and his PhD also in molecular and cellular biology. Then he moved on 2001 to London to the MRC Imperial College, work at the lab of Luis Aragon, where we met because we were both doing a postdoc at the same building. And he did a terrific work there, working on the segregation of the ribosomal DNA during mitosis. And in 2005, he obtained a Ramón y Cajal contract and came back to Tenerife to start his own group, where he is now at the Hospital Universitario Nuestra Señora de Candelaria. And there, Félix is doing a very, very interesting work also on DNA damage and chromosome segregation, very interesting things with the role of CDC14 and topoisomerase II on anaphase. And very most recently, and I think is what he's going to speak us today about, on the important role of the reconfiguration of nuclear morphology for DNA repair. And well, just to say again, thank you very much, Félix, and we are looking forward to your talk.
So thank you very much. I hope that you can hear me well, all of you. Is that correct? Now I need to figure out, okay, I need to share screen. So can you see the presentation?
It's perfect.
Okay, so, okay, Maria, thank you again for your nice introduction and for inviting me to give this talk as part of your group with the society. And well, you've already said many things, so I don't need to introduce myself again. In this first slide, I'm just going to show you the title of the presentation. I need to find a second. Here we go. Which is Beyond the Sphere, the nuclear shape in chromosome structure and DNA repair. I must say that I'm going to divide this talk in two. One which is related to chromosome structure, and the second part which is going to be dealing with DNA repair. In this first slide, I would like to acknowledge the organizations I am part of and the funding agencies that have supported my work for so many years, national and regional funding agencies. And without any further delay, I would like to introduce you all. Before I actually talk to you about the topics of today, I'm just gonna tell you a little bit about what has been probably the main line of research in my group since I started my postdoc and after I came back here to Tenerife, which is, I think it's important to relate what I'm gonna talk about later, although it's not exactly the same thing. But anyway, this has been one of the main topics. I think it's going to be easier if I put the pointer here, which has been I've been interested in knowing the causes of underlying this strange aberrant phenotype in anaphase, which are called anaphase breaches. As you can see here, is when cells try to pull the replicated chromosomes, which are then called sister chromatids, towards their daughter cells, sometimes there are problems which are going to compromise seriously the genomic integrity, and they are presented, for instance, in this way, which are called anaphase breaches. So this aberrant phenotype in anaphase has been one of the main topics I've been interested in for many years. And from a conceptual point of view, they could arise from either polycentric chromosomes, this is chromosomes which have more than one centromere, or are problems during the resolution of sister chromatids. This is after you replicate chromosomes. Again, from a theoretical framework, you can have different sources of these problems that could lead to these unresolved sister chromatids. For instance, the presence of proteins, linkages between the sisters. The most obvious candidate for many years have been the cohesin. If you don't remove cohesin, which is what maintains sister chromatids together until anaphase, and then topological issues in the 3 dimensions, such as the catenations and knots between the 2 sister chromatids, or direct linkages in the DNA, such as unfinished replication. So imagine that you enter or try to enter anaphase before you have finished replication, or the presence of recombination intermediates, which are part of the in the process of DNA repair, arise as a consequence of cells trying to repair the DNA, and then you can have this kind of intermediates that could interfere also with the segregation process. And I've been using yeast as a model system. So yeast has 16 chromosomes, thereby it has 32 chromosome arms. And I've been interested in trying to focus on one single chromosome arm, which is the right arm of chromosome 12, because it carries what is called the rDNA, or the ribosomal DNA array, which is sort of the most repetitive region in the yeast genome, and is very long compared to the rest of the genome, and makes this arm the longest in the genome. As you can see here, this is just a measure of the arm length in megabases, and it's clearly the longest one of all. But aside from this length, you also have certain particularities about this ribosomal DNA, because it's formed of, well, in around 150 copies in a head-to-tail tandem array, and they are highly transcribed. It's probably the most transcribed region in the genome. It needs like devoted RNA polymerases just for one single gene. For instance, I guess the RNA polymerase 1. That's the only job that this polymerase does in yeast. And because of this high transcriptional level, it has many problems during replications. For instance, a collision between the replication forks and the interspersed transcription machinery. Is a heterochromatic region. Is known to be hyper recombinogenic. Is prone to be entangled, have all these topological problems. In fact, it's a region which is enriched in condensin, a protein that probably maybe some of you already know. It's important for chromosome condensation, but also for resolution of histoachromatids. And also it's enriched in topoisomerase 2, which deals with catenations. And in fact, when we started studying the segregation of this chromosome arm, we took advantage of several things. For instance, one important thing is that this region, because it's particular, and there are many proteins which bind to the regulatory elements throughout this array in every single copy, and they only bind to this region, not anywhere else in the genome. So you can have a specificity to recognize the rDNA just under the microscope. If you tag one of these proteins, here you got 2 examples, which is FET1 and FOC1, with fluorescent proteins, and they are bound strongly to this promoter. So it doesn't matter. So they are always bound here. It doesn't matter whether you go through different stages of the cell cycle. If you stress your cells, they are going to be always there. And aside from these specific proteins that code the RNA, you also have like what we call a system which is based from bacteria, TET operator, and TET repressor system. So the TET operator is a sequence that you obtain from bacteria, and then you can integrate anywhere in the genome, the region that you wish. And then using the TET repressor, which is specific binding protein for the sequence, tagged to a fluorescent protein, you can recognize these sequences as spots. And when we did this, using different strains with different locations for these spots or combination thereof, what we could see is that, first of all, this region in normal wild-type cells during anaphase is last to segregate. So this is the chromosome R that it takes more time for segregation. So you can have like the mass of the nuclear masses, as you can see here, stained by DAPI, completely segregated, but you still have like sort of transient bridge of the RDNA connecting both. And when you go to the fine detail of what this bridge looks like, it's sort of like this. So the segregation starts from the centromeres, pulling the centromeres away, and then in a sort of HUNSC mechanism, you go from the centromere to the telomere, and then you end up resolving the telomeres. And there are a bunch of mutants, a bunch of proteins which are important for these last stages. And this is so because there are many mutants, the cells get stuck in this pattern, with this abnormal pattern of anaphase bridge. And among them, we identified many years ago, proteins such as the master cell cycle phosphatase CDC14, condensing, another related complex which is called SMC56, topoisomerase 2, and under DNA damage, also the structure-specific endonucleases that resolve these sort of linkages I showed in the first slide. So, which means that this is a challenging region for segregation, and you need cell factors, which are important to give you the last push for the segregation, so to speak. So aside from these causes of trying to uncover the causes of these RDNA bridges, one thing that I've done also in my lab is having this model of anaphase bridge restricted to just a single chromosome arm out of the 32 chromosomes arm of yeast. We've used this to see what happens after you enter a new cell cycle. After you try to enter a new cell cycle, if you force cells to perform cytokinesis, probably what's going to happen is just you create these double-strand breaks, which are quite particular because these ends cannot be bring together anymore because they are in different compartments, the daughter cells, and also they are symmetric. So in haploid cells, that you are not going to have anything to repair with, for instance, through break-induced replication or other forms of homologous recombination for these cells, whereas in this one, maybe you can still use the sister chromatid, which is intact for repair. So we wondered things such as, well, what happens to these cells in the short term, what happens in the long term, what happens for cells which survive, and using diploids and genome-wide assays. We also, with the gene, I must say that the diploids is much more complicated. It's just showing here things because it depends whether you've got one of the homologs with this bridge, you've got both of the homologs, and depending on which way each of the broken sisters are going to be segregated, you can have different ways to survive by recombination or by other means. Well, it's something that we have studied here. So, but then suddenly I move a little bit. I just told you I've been interested in anaphase and what happened after anaphase in the next cell cycle. But suddenly, for this work, the first work I'm going to present today, I move a little bit earlier, right at the step before anaphase, in metaphase. And why? And this is what I'm going to talk in the first block, which has been published. So it was published 1 year ago, around 1 year ago, and recently we just published a review with some insights and perspective about this previous work. And I was interested in this for a long time. because the rDNA had been used before as a model system to address chromosome structure in Saccharomyces cerevisiae, because it was the only region, because of its repetitive nature, that by using fish, you could follow the chromosome structure in yeast, because in yeast, when they get to mitosis or into metaphase, they don't look like animal chromosomes in animals or plants. You don't have like a, they still sort of amorphous morphology. But it doesn't happen only for the rDNAs, where you can have a sort of like a structure, proper structure. So in G1, you've got sort of a dispersed array if you use fish as a technique, and then suddenly when if you block cells in metaphase, then you've got this sort of bent bar or line structure, which was referred to as a loop. Sometimes I'm gonna call it a horseshoe loop because it looks like a horseshoe, or maybe a handle because it also looks like a handle, in this case of a cup, or maybe a handbag here. And it was called an organized structure. Actually, it was called a condensed state of the rDNA, because this work, which was pioneered by the Koshland lab, they showed that important structural and maintenance complexes such as cohesing and condensing were very important for the establishment and or the maintenance of this structure. And because condensing it was so involved in the formation of this structure, it was referred to as a condensed state of the chromosome. And the funny thing about this is like that this is something that we can also see with our system of specific proteins that code the rDNA, as you can see here. But there's a big difference if we compare this to fish. With fish, you've lost a lot of information, because for fish, you need to process a lot your samples. But in our system with fluorescent proteins, you still can put this in the context of the whole cell, in the context of the organelles, in the context of the membrane system that you can use yourself. And one important thing also is that you can follow this in live cells, cells which are alive. For instance, you can perform video microscopy. This is just an example. In this case, you've got a crowded population of cells which are arrested in mid-M, this metaphase state with indocodazole, which has been on the standard procedure for arresting cells in metaphase. And this is very crowded, but every cell of this looks like this. But I just did it this way because we wanted to have as many nuclei as possible for just a single small field. And you can see here 15 nuclei, just static moving of 13 minutes in localosol, cells already arrested in localosol. And you can have, well, you see all this loop as it can be seen from different visual perspectives, from views, top views, maybe this one is a top view, or side views, maybe this one is a side view. Most of them are top front views or slightly tilted views from this front view condition. And the things, I would say that what called our attention a lot were three things. The first two things is, or the first one is just like, if you put it in the context of the whole cell, or the mother cell, which is this cell here, this nucleus, so all your DNA is here, and suddenly you've got this loop. It's very impressive, and then you've got this in here. But if you consider that this is the whole nucleus, this nucleus defies one of the laws, or one, I could call it a law in biology, which states that there's a ratio between the nucleus and the cytoplasm, by which the nuclear volume cannot be bigger or larger than 10% of the cell volume. And this is present in all kingdoms, in all cell types, except for cancer cells, where you break this rule and you got massive nucleus in the cell. So, and it looks like, apparently, that this is breaking the rule as well. The second thing is that I told you that Koshland defined this as a compaction or condensed state of the rDNA. but also in their works, they establish what is considered the compaction ratio of the general chromosomes, these chromosomes in a metaphase rest, which is 140. This is, chromosomes look like 140 times more condensed from the longitudinal point of view than the naked B-form DNA, right? If you measure the length of this RDNA in this condition, it's actually 80, which is less condensed. So this state is probably a less condensed state, at least from a longitudinal point of view, than the standard or state of metaphase chromosomes. And one important thing that we realized at the very beginning is like, if we took one strain, I told you that this is normally made of copies which are repeated, at the normal cell have like 150 copies, and it's maintained, dynamically maintained. But there are tricks through mutants where you can actually fix the number of copies and you can make it smaller, as you can see here. And when you do this, you can go up to 25 copies. If you go less, then you got a serious problem for growth. But with 25 copies, cells are more or less still like a wild type strain. And what we found is like the length of the RDNA was still the same. So it was constant. We were expecting to be variable, but it was actually constant. What was variable was the condensation then of this. So you reduce the longitudinal compaction. And this leads us to the final thing that caught our attention, which was the space under the loop. So it was, this spacing here was constant, even when we decreased the number of units of the RDNA. At the beginning, we thought that this space here, what we call it SUL, space under the loop, was basically an enlarged nucleolus, because cells were arrested in metaphase, they are still producing ribosomes, ribosomal particles, they are transcribing the ribosomal RNAs, and so forth. But to make a long story short, it turns out to be non-nuclear. So if you use a protein, which is in this case is just a free pool of a TER repressor, so we got the strains where not all the TER repressor which should be bound to the TET operator. So you've got a pool of TET repressor which is not bound because there's too much TET repressor in these strains. So then it labels, because it comes from bacteria, doesn't bind anything in the nucleus, so it stains the nucleoplasm. And as you can see here, when you've got this loop, then you've got the void space here. It's not nuclear, so nucleoplasm cannot reach this space in the middle. And I'm going to show you here like a movie, which is actually a reconstruction of a cell, where you're going to have three markers. What you're going to have in these three columns is a work through the different set slices reconstructed through confocal super resolution microscopy. And in the last column is going to be a 3D reconstruction rotating on the y-axis. And then for each of these columns, you're going to have like two markers, combination of two markers of these three. So the first one in red is going to be the chromatin. The second one is this net1, which is going to label the RDNA. And the third one is a marker for the nuclear envelope. And if you see this movie, then you can see how there's nuclear envelope. Of course, there's nuclear envelope all covering the whole chromatin. But there's, for the RDNA loop, there's nuclear envelope, of course, on the outer face, as you can see here, but also in the inner face. I hope that you can see this very well now here, right? So it's clearly non-nuclear, this space. But if we if we look also through this TET repressor, many times what we could see is that there was a constriction when you got this donut-like nucleus or toroid nucleus. There's a constriction, a terminal constriction here, which is here indicated by these white arrows. And as if this was made of a nucleus which has like two lobes. one lobe here, which is number 1, which doesn't carry the rDNA, but I cannot show you here now, but that is where most of the chromatin is, chromosome masses are, and a second lobe which is just the distal part, and where the distal part of the rDNA is gonna be. So it's made of 2 lobes. And this is something that we also observed when we filmed. In this case, it's a time series, it's a movie, which is a time series, where you can see how this spectacular morphology of the rDNA loop arises. So cells are after you add nopodazole, and they are 2 examples taken from cells which have had a small bud at the beginning, which were it means that they were in earliest phase, and they progressed through metaphase. And you can see here how this round nucleus, suddenly there's a finger arising here, finger of membrane arising where the rDNA is, and this finger grows, and suddenly becomes thicker at the end, forming probably this sort of bilobed nucleus where the 2 lobes are touching each other. And to make things even more complicated, in about half of these mature donut-like nuclei, if you go and do a series again slices, what we noticed is like the inner face of the nuclear membrane closes in onto itself, which means that if we reconstruct this in 3D, what we have is a nucleus like this, with what we call a ladle, or looks like spoon, with the rDNA being the edge of the spoon, and we probably the 2 nuclear envelopes, the bilayer, because the nuclear envelope is made of two bilayers, the inner nuclear membrane and the outer nuclear membrane. Probably the 2 inner nuclear membranes very close to each other, so that you don't have space for the nucleoplasm to transit freely. So what's in this space then? What's in this space? What's the whole of the donut, I could say. So to our surprise, when we label cells with a cytosolic GFP, we couldn't see the cytosolic GFP in this space either, which was quite surprising. So it's not nuclear, it's not cytosolic. What's in there then? Well, from bright field images, it was relatively easy. It's something that actually we noticed even before we did this experiment, is that we got this refrigerant sphere here, which is normally correspond to vacuoles, something that is pretty known for this biology. It's a classical pattern of visualization of vacuoles by bright field. And this is again another picture. In this case, it's sort of like tilted, slightly tilted from view of a loop, and you can see here like the vacuole is sitting on this space under the loop. And this is just a 3D reconstruction. We're rotating the y-axis. In this case, using the histone, because also with the chromatin, you can see also this handle here, which corresponds to the rDNA. And in this case, the vacuole is just a vacuole membrane protein, which is labeled in green, VPH, and you can see how this is sitting on this space. Well, you may think, okay, this is not as strange as it seems, because the vacuole is large, the nucleus is large, so maybe just by chance this takes place many times. So this end product, but it happens to be, it turns out, that this relationship, as you can see here, it starts from the very beginning, from the very birth of this finger, like projection. You see always the vacuole like this, and the finger sort of wrapping around the vacuole. You couldn't see this kind of figures in a time course in nopodazole. As you can see here, almost always, it was next to this finger. So this is just a brief summary of what it depends on. It's just a mechanistic point of view. We demonstrated that this donut-like nucleus and this outstanding rDNA loop depends on the active production of nuclear envelope, and this relates to previous work by from Orna Cohen-Fick's lab, where they already demonstrated that under a knockout-sol arrest, nuclear envelope production over expansion is unabated, and mysteriously all that new nuclear envelope goes to the nucleolar subdomain of the nuclear envelope. So everything sort of makes sense. This is something that we demonstrated here by inhibiting fatty acid production before arresting cells in metaphase, and then you don't have this nuclear anymore. This case is done by sirtulinine, which is an inhibitor of this enzyme. We've done it also through more genetic approaches. And if you play the other way around, so if you knock out, you interfere with the phospholipid with the lipid synthesis, like pushing towards the overproduction of nuclear envelope, even in conditions where you could have sort of an equilibrium between lipid droplets and endomembranes. As you produce more endomembranes, then you can have the formation of these loops still bound to the back wall, as you can see here, even in cells which are not arrested in metaphase. So these cells are basically in G1 early S phase, so you don't need this arrest in metaphase any longer. And just finally, this is something that actually comes from a previous work that we did, and that where we show that this loop depended on TOR. So if you create or you stress your cells in conditions where you're going to inactivate TOR, then this loop suddenly disappears. So it can be reversed, this phenotype, and this is something that we did here. This is by measuring the length of the loop. And then even with rapamycin, which is a proper inhibitor, a specific inhibitor of TOR, you can have this. To put all together, what we think is like in a round nucleus, which is a normal situation in asynchronous cells, if you block cells in the cell cycle, what you've got is that this nuclear envelope continues growing, because probably needs to grow in order to segregate chromosomes in anaphase. But because you don't have anaphase anymore, so it looks like it makes a different pattern. Makes this sort of bilobed nucleus with this ladle here with a vacuole in the middle, and it needs like an active TOR, because even in this no progression of the phase, you still have an active TOR. You've got this nuclear synthesis, and then what happens is, like, if you suddenly inactivate TORs, so it happens that maybe you reverse this phenotype, and we don't know whether this happens because maybe during activation, what you do is like maybe you upregulate autophagy, and autophagy is somehow dealing with this excess of the nuclear envelope, or else because it's known that after you inactivate TOR, you are going to stop transcription by RNA polymerase II. And although I didn't mention this, if you stop transcription, then you've got condensation of this rDNA array. This is known, that happens in anaphase, normal cycle, and is something that we are foreseeing for future studies. So in the second part of my talk, I'm going to talk about a sort of different thing, which is related to DNA damage and DNA repair. And we'll see that as it converges into something similar to abduction. Okay, but the reason behind this study was, well, yeah, just going to give you a brief introduction of double-strand breaks. So you know that double-strand breaks are very serious DNA damage for the cell. So there are different insults, endogenous or exogenous, that are going to break the DNA. And then you got two ends that need to be repaired. There are two mechanisms, basic mechanisms. One of them is non-homologous enjoining, where you basically take these two ends and put them back together. But it's known that this is mutagenic, because normally when you create these DNA double-strand breaks, you need to process these ends before they are ligation. They are able to ligate again, and then you create normally microdeletions. This non-homologous enjoining is normally preferred when you don't have a sister chromatid, which can be used as a template for the second mechanism I'm just going to introduce to yourself soon. And this is a mechanism that normally works in G1 cells, and is regulated. So it's better, or it's promoted when you've got like low levels of cyclin-dependent kinase activity, what happens in the normal situation in G1 cells. When cells are in S or G2 phases of the cell cycle, then you've got high CDK activities, which is needed for the progression through S phase and G2. And when you do this, you actually upregulate this alternative way of repair, which is much more complex, which is called homologous recombinations. And this is done because cells sort of know that they now have a sister chromatid, which is a reliable template to be used for the repair in order to seal this broken molecule. It's a very complex process. I'm not going to get into details, but it's very, if it's done properly with the sister chromatid instead of other ectopic sequences of homologous chromosome, it's quite safe for the genomic stability of the cell. So, but there's a paradox for this system. So if you go through the literature, everyone says, oh, it's very clever. So you go low CDK activity, you promote non-homologous enjoining. As the CDK activity rises, then you go for homologous recombination. Very clever, very clever. And as soon as your CDK activity drops again, in G1, then you go for non-homologous enjoining again. But there's a paradox for this system here, because in mitosis, actually in anaphase, you still have high levels of CDK activity. So the CDK activity only drops during right at the transition between telophase and G1. But after anaphase onset, you don't have sister chromatids anymore aligned to each other. So they have been split apart because they have been segregated. So you got high CDK activity, which should promote homologous recombination, but you don't have the template for homologous recombination any longer. And this is the paradox. And this was the paradox we were interested in the past. And then we realized, because one good thing about yeast is that unless mammal cells, is that you can arrest cells in late mitosis as well. So there are mutants where you can arrest your cells in this stage with your nuclei segregated. Just a reminder that they undergo what is called a closed mitosis. They don't break down the nuclear envelope. And they look like this. So if you are arrested in telophase, we did it using a thermosensitive allele for this protein, CDC15. So we shifted the temperature to 37. And after 3 hours, you've got this nice arrest in late mitosis with all your chromosomes segregated, including the RDNA in this case. And then if you create DNA double-strand breaks in these cells, what surprised us a lot was that we could see a reversion, a regression of anaphase. So there were like de novo, retrograde anaphase bridges forming. This is something that you can see here. This is a quantification of these figures, which were not present at the arrest. And if we follow like specific sister foci, we should be segregated before or during the mock treatments in this arrest. What we found is like these sister foci sort of go back to the mother or to the daughter and coalesce again. They are brought together again. And from a clonogenic point of view, if you measure how efficient cells can recover from DNA double-strand breaks in G2 or in G1 or in telophase, what you can see here is like in G1, because you only have non-homologous joining, they are quite hypersensitive to that double-strand breaks, and they are less sensitive in G2M, because then you can repair with homologous recombination. And indeed, if you knock out homologous recombination, which is something that you can do for knocking out this essential gene, which is called RAF52, then you can see this drop in the viability of the cells, right? This clonogenic axis. But in cells arrested in telophase, as you can see here, they more or less look like G2M. And they are also responsive to this mutation in FAT52. So it seems that these cells, despite they have segregated the chromosome, they are still rely a lot on homologous recombination. So it seems that homologous recombination is still working here, and they do it by bringing back or sort of, bringing back chromosomes after they have been segregated. So the purpose of this work was actually trying to look for specific factors which could be important in homologous recombination or in double-strand break repairs in late mitosis. And what we did was a proteomic assay. We've done different approaches for this, but the one I'm going to show you here is just a proteomic assay. So we were looking for proteins whose abundance, this is the abundance proteomic, change after the induction of double-strand breaks in cells which are in telophase. And we did it with a mock treatment, no double-strand breaks at all. And then we used two different kinds of double-strand breaks. One, which is the pleomycin, this compound, which is going to give you like randomly distributed double-strand breaks, and one which is an endonuclease, which is going to clip a single region in the genome. And when we compare this and when we compare this in other conditions, for instance, in G2M, looking for these specific proteins which change it in late mitosis, we found this protein, which is called MSC1, which is a really ill-characterized protein. But funny enough, there's a paper in the late 90s where this protein was actually identified in screening for proteins which were important in meiotic homologous recombination and the discrimination between the homologs and the sister chromatids in meiosis. But nothing else has been done so far since. So we validated these proteomic results by Western blotting, showing that this protein was enriched specifically after double-strand breaks in late mitosis relative to G2M in both conditions with both type of double-strand breaks. We also confirmed this by microscopy. So there was an enrichment in intensity of this protein in the cell. By the way, importantly, this protein is a nuclear envelope protein. As you can see here, it labels the nuclear envelope, and this is going to be coming closer to what you are going to find out soon. And intriguingly, it was unevenly distributed in the nuclear envelope, and it looked many times, not always, but about 25% or 35% of these late mitotic cells, there was enriched in one or more patches on the nuclear envelope, as you can see here. But didn't change, although it's not working my mouse anymore. I don't know why. Okay.
We can see the mouse moving, but maybe it's not.
I'm just gonna use. I don't know. I don't know what happened.
It's working. It's working properly.
It's working, but it's not getting where I
for the next slide. No?
I want to. Yeah. So yeah, what's important here is like didn't change these patches. The percentage of cells present in these patches didn't change after we created the double-strand breaks. It was present before. If we knock out this gene, what we found is that indeed was hypersensitive to double-strand breaks, was hypersensitive to pleomycin. As you can see here, this is just a control, the FAT52. It's important protein, but if you compare this to the wild type, the mutants, the wild type is clearly in this spot acid that was more sensitive. And it was also more sensitive when we induced this HO, this double-strand break created by an endonuclease. I must say that endonuclease is expressed under the control of the beta-stradiol promoter. That's the reason that you see here, is beta-stradiol. I don't know why the mouse is not working. But it cannot get. Anyway. We, as I said before, we were interested in knowing whether this was also more specific to a specific stage in the cell cycle. So we cannot address that by classical spot assay. So what we did instead was a clonogenic assay of survival, as I showed you before for in the previous slide. So relative to MOB, what we found is like this mutant was more hypersensitive after double-strand breaks, both double-strand breaks, when they were induced in cells which were previously arrested in late mitosis, relative to G2M, which it seems it also drops in G2M, but it seems that the penetrance of this phenotype is stronger in cells which are previously arrested in late mitosis. If we go for the mechanism, I'm just going to briefly say that the first thing that happens after double-strand breaks is that you put sort of a sensing mechanism that is going to sense the double-strand breaks and block cells and prepare cells for their repair. And this is done by a sensing protein which is called RAF53. And it's a way to measure how much proteins, how much cells are sensing DNA double-strand breaks, because this protein becomes hyperphosphorylated. The more hyperphosphorylated RAF53 gets, the more signal, the more presence of double-strand break you put. And as you can see here in this time course of the double-strand breaks, if you compare the wild type and the mutant, it seems that the mutant is sensing more double-strand breaks. Of course, we don't think that there are more double-strand breaks in this mutant. What happens perhaps is that these double-strand breaks are not repaired as efficiently as in the wild type after you induce the double-strand breaks. And that's the reason that you still have a higher presence of this reporter for sensing double-strand breaks. And indeed, when we perform this assay, I'm not going to get into details again about this assay, but it's called the MAT switching type assay, this classical yeast assay to address double-strand breaks. Just focus on in this triad of bands. So what happens normally is that you create a double-strand break in this MAT region with this endonuclease HO that you induce with a beta-stradiol promoter, and then this is going to use it's going to undergo an intramolecular recombination with this region here, which is called HMAL, and which is going to give you a gene conversion, which means that the product of the homologous recombination is going to be identified by a fragment length polymorphism. In this case, based on the presence or absence in the parental and the template of the ST1 enzyme. So what happens is like you start with this band here in a southern blot, then you go to a smaller band here, which is what you get after you cut with the HO, and then this band disappears after this cut is processed, this process. And if you go to a non-homologous enjoining, what you do is that you will restore this MAT-A, but if you go to homologous recombination, you instead you get this MAT-alpha band. As you can see, all in this late mitotic arrest, everything goes through homologous recombination, nothing goes through non-homologous enjoining, and you get to this band as a final product. But the difference between the wild type and the mutant was the time that it takes to get to the final product, and there was a clear delay in the repair process in this mutant. So it seems that this mutant has a problem with the repair. And accordingly, another way to measure or to see repair by homologous recombination cytologically is by looking at RAF52, because RAF52 and other proteins involved in homologous recombination, they form what is called repair factories in the cell. So if you look at cells which have been exposed to double-strand breaks, they form foci. And if you measure the foci that you see that cell, then after you create double-strand breaks, where you have more foci, as you can see here, with HO and cleomycin, after 1 hour, after 2 hours, you get more and more foci. And after they are repaired, you get again fewer foci. But in the mutants, clearly, in the mutant, you can see there are fewer foci, always fewer foci, relative to the wild type. So it looks like it takes, so the cells struggle to repair double-strand breaks in this mutant. And as I mentioned before, there's a patch. And when we look at cells, this a little bit complex graph, but if you just pay attention to cells where you already have either one patch or one Raf52 foci per cell, and you see whether this colocalizes in the same .2b nuclei, or this nuclear body in this elongated nucleus, the mitotic nucleus, what you see is like a 3.5 ratio in favor of Raf52 colocalizing with a single patch, a single Raf52 colocalizing with a single MSC1 patch. Of course, in this one, it is a wild type with this protein label, which means that there's a correlation between the patch that we observed before, although it didn't change after double-strand breaks, it actually correlates with the presence of the Raf52 foci. So we thought, sorry if taking a little bit longer than expected, but yeah, I'm just finishing. I'm just gonna mention what this protein is. So, so this protein, I haven't talked about it. As I told you, it's an ill-characterized protein. But what is important about it is that it has a single, as I showed you, it's a nuclear envelope protein, but it has a single transmembrane domain, which is very close to the N-terminus. It's so close to the N-terminus that you cannot say that there's anything here functional. So everything which is functional in the protein is here in this C-terminus. This protein doesn't have homologs out of the kingdom of fungi, which is a problem. But in Schizosaccharomyces pombe, it has two homologs, which they look like the same, more or less the same. And it carries a motif, which is called EISH1 motif, because it was first discovered in this protein, which is one of the homologs, but nothing is known about this motif. Actually, very little is known about this protein. And in Saccharomyces cerevisiae, nothing. And in pombe is a little bit more known about it, but not more. But what's important is like, okay, if you have a nuclear envelope protein and it's doing something with the homolog of recombination, and it's a nuclear envelope protein, of course, and then you got these regions such as single finger like motifs here, and as has been said also, that this ISH1 motif could actually also bind DNA. Okay, what you got is an inner membrane protein facing the nucleoplasm, which is sort of tethering maybe the chromatin, because it's known that tethering the chromatin is also important for DNA repair. But a paper a year ago in the two homologs in pombe showed that this part of the protein is actually facing the lumen of the nuclear envelope. So we wonder whether we had the same pattern here. Just this is the system we used. They did it through electron microscopy. We did it through a system which is basically relies on la reconstitution of functional GFP proteins. So what you do is just you tag your protein with half of the GFP protein, and then you tag the other half of the GFP protein to different reporters that are going to locate in different positions along the nuclear envelope. So it could face the inner face of they could be in the inner nuclear membrane facing the nucleoplasm. There could be either in the inner nuclear membrane or the outer nuclear membrane facing the lumen, or could be in the outer nuclear membrane facing the cytoplasm. And what we found is that this protein is facing the lumen. All the C-terminal region is facing the lumen. Nothing is facing the nucleoplasm. And well, one may think, okay, but this is maybe the situation before DNA damage. Maybe this protein could transverse, could translocate, and may faces the nucleoplasm after DNA damage, but it doesn't. So it still faces the lumen. So we could only reconstitute the GFP protein in the strain, which is a reporter for the protein facing the lumen. So this means that it's not as straightforward as we thought. And this led us to this other paper. This other paper is a Nature paper published 2 years ago with the LES1 protein. Is the second homolog in Schizosaccharomyces pombe, and it's important because it was the first protein which was actually involved in regulating karyokinesis. And basically, what happens is like without this protein in this mutant, you have sort of like have a mess for karyokinesis. So there's leakage in the nucleoplastic bridge, and a schedule and tiny karyokinesis, etc. And of course, we said to ourselves, oh, clearly that's fine, because if we have a premature karyokinesis in our situation with blame mitotic, in this mutant, of course we cannot repair these double-strand breaks anymore with homologous recombination because they cannot bring together anymore. But it turns out to be that the influence or the in karyokinesis of this mutant is very little, if so. So we couldn't see any significant difference in karyokinesis. What we found doing these assays instead is like this mutant tends to have like what we call compartmentalization of the daughter-to-be nuclei. So the daughter nuclei, instead of looking like a dumbbell or our glass, like with one body here, a bridge here, and one segregated body here, they appear like bilobe, like segmented in different with like septums in between. And this is something what you can see maybe here better in this reconstitution of slices. and it's nuclear. It's not just because we can prove it, because we also see this sort of septum with nuclear pore complexes, not only with this marker for the nuclear envelope, but also with nuclear pore complexes. And this septum actually it sort of compartmentalized DNA. So the segregated DNA is now found in like in different compartments. So, and finally, if we look at movies of these patches, I'm not going to show you a movie now. What I'm going to show you is a time series of one of these movies. And we look at these patches of MSC1 protein. What we found is that mostly these patches appear in regions which appear as plebs in your nucleus, or steps which could be prior to the septation of this compartmentalization. And you can see here how these patches sort of resolve or solve this problem. Although it's quite dynamic because it comes back here, sort of comes back here at the end of the movie. But if you go from the beginning where there's a bleb here, you can see how this is repaired, and this nucleus sort of becomes rounded again, becomes like more spherical again. So our working hypothesis now is that this mutant is important for homologous recombination because prevents abnormal situation, abnormal configuration or reconfiguration of the nuclear shape in late mitosis, which could trap the sister chromatin, which is used as a template for a late repair of double-strand breaks through this process of approximation of sister chromatids. And maybe because it traps the sister, one of these compartments cannot any longer reach this sister, and that's the reason that cannot perform homologous recombination. And with this, I'm just going to finish. I'm just going to acknowledge my students that have done all this work. So for the first part of the work of the talk, the work with the vacuole and the restructuring of the shaping of the nucleus in metaphase arrest, just basically the work of Emiliano Matos Perdomo and Silvia Santana, and the work with the DNA damage and this MSC5 protein has been done mostly by Sarah. Well, Jessel has participated in both of these works, in early stages of this work. I'm happy to take your questions.
Thank you very much. Just remember that you can ask your questions in the chat, both from YouTube and also for the Zoom. Thank you very much, Felix. Very nice talk. So I think that till now we don't have questions.
Hi, sorry, can you hear me? Because
yeah.
Oh, okay. Right. I wasn't sure if I could. Oh, thank you very much, Felix. Very nice and amazing microscopic images for such a small nuclei. I'm very impressed. I can start. Meanwhile, I have a couple of questions, mostly of things I am not sure if I understand. one of each part. So I can start from the second one. This is pretty amazing, no? So it is MSC1. So you conclude that it's required to prevent this segmentation of the sister chromatid, and so it's not from a homologous recombination per se. It's just to facilitate the template to be ready for the other sister chromatids. That's what you think. And is that why you think it's only occurring, or it only appears in organisms with closed mitosis?
Yeah, maybe. I don't know if you can still see the presentation. Is it on? Well, okay. I didn't mention because, of course, I wanted to go faster because time was running out. But what I didn't mention, as you say, is like this. So these problems are before, if they are in the mock experiment. So this compartmentalization is before. It is happening before. Actually, we induce the double-strand breaks. And again, as we could see for patches, there's no difference. So you don't have like a more compartmentalization after DNA double-strand breaks. It's before. So what we think is that this happens before. So and that's and then you have all these. You have all these aberrant late mitotic phenotypes, and then maybe after you induce double-strand breaks, because we see this already by DAPI, we see this sort of split in different masses. DAPI more than 2. It should be just 2 for one daughter to be nucleus or one daughter to be nucleus after you. But then you have sort of like more loads in your segregated nucleus like this, that is depicted here. And the thing is that maybe if DAPI wasn't here, or maybe you think, okay, this is something. But when you find DAPI like split between these different compartments, the conclusion is that maybe it's much more difficult for your double-strand breaks to find the right plate sister chromatid if you reverse this segregation. I don't know if I answered your question.
Yes.
So, and then it's true. I mean, we think that this is important for closed mitosis. Yeah, definitely. It's something that I would say is something that has not been explored. Maybe think, oh, well, you're looking at closed mitosis, it's not the same. But just even if this cannot be translated into mammal cells, right? Still, closed mitosis is something that takes place in many pathogenic organisms. Yeah. So not only yeast, and even for pathogenic yeast, the candida and all fungi, many protists. So understanding how cells respond to double-strand breaks, DNA damage, maybe it's important. Maybe if you want to play, do you want to play maybe with combined therapies that could be useful for this? So maybe creating double-strand breaks, but at the same time interfering with closed mitosis. Closed mitosis is a target. I would say that it's definitely a target that should be explored.
Yeah, because it's unique. Yeah, but do you think then that it would have in either in other parts of the cell cycle some role in the nuclear formation, or I don't know. I mean, because what I meant before with my question is that you see this phenotype in double-strand break resolution because that's what you are saying, but seems to be physiologically relevant to have proper nuclear envelope formation. So do you think it can have?
Well, the mutant is only, I mean, apparently grows like the wild type. In growth curves, it's a little bit slower than the wild type, but not much. In spot assays, etc., it looks like the same, colony size, the knockout mutant. So clearly is not essential. So somehow cells deal perfectly with these problems. So somehow they manage to survive.
No, but maybe if you challenge the mutant cells with something else, not only DNA damage, then you will see some of the phenotype.
Well,
you would expect.
We don't know. I mean, we haven't, I mean, with oxidative stress, for instance, you don't see there's something that we did. We also, because it was previously reported that this mutant could be important in copper resistance in one of these omics assays, but we did spot assays with copper, and we couldn't see any difference between the wild type and the mutant. So for oxidative stress, you don't see any difference either. So it looks like only so far we haven't seen it only for DNA damage, but it's true that is the only thing we've mostly, aside from oxidative stress and copper, is the only thing that we've tested this mutant against.
Now, this is really interesting to check the synergism with different therapies in order to use this in pathogenic yeast, but it would be amazing to have some similar mechanism in mammals, huh? Really, because
yeah, I mean, I'm not saying that this is a substitute. I just want to be cautious about this.
Yeah.
But the phenotypes that you observe sometimes is the phenotype sometimes that you observe in lamin deficient cells in mammals, and it doesn't have a lamin.
Sorry, with Felix.
Lamin, but I don't know if lamin is like.
Yeah. Yeah. It's really interesting. Really interesting. And in the case of the other part of your talk, maybe I lose something because I'm not expert in this topic. But do you think, or do you have any data demonstrating that the back wall is doing actively something in the DNA that is surrounding this back wall, and which is the content of the back wall that could be involved in this regulation? Do you know something about this, Felix?
Well, we think, as I yes.
Because you see that is the condensed, no? Maybe the
gradation. It's sort of be stretched, I would say, stretched, and okay. We know it's like depends on TORC1, for instance, and then suddenly you don't have. Well, you don't have. I just wanna stress that for the TORC1, the only thing that we have properly done is measure the RNA. So it's something that we should need to explore further in terms of what happens to the membrane. Because maybe what happens is that you got like a condensation of the RNA, but maybe you still have the membrane. We've got some data actually saying that there could be still some membrane here. So maybe this TORC1 goes more through this condensation of the RNA after you repress RNA polymerase 1 transcription. But it's intriguing that you see the vacuole here. So this close, intimate like relationship between the expanding nuclear envelope and the vacuole. The one thing I didn't show you, because is again for this look, it looks like it's sort of independent of the connections of the non-junctions that occur between the vacuole and the nuclear envelope. But there are a bunch of junctions. But the most important junctions, which are the ones related to autophagy, nucleophagy, or several pathways of nucleophagy, and also the ones which relate to lipid droplets, synthesis, and the nuclear envelope. It looks like it's independent of that. So we've got some data suggesting, but this is something we haven't proved, suggesting that there could be a difference in the composition of the lipids. So the lipids that you find in these overextended membranes could be different from the lipids that you find, or for lipids that you find normally in the nuclear envelope. And so maybe that's this underlying cause of this interaction with them.
Really interesting. We have a question here in the chat from YouTube. It's from Pedro Antonio Sansegundo Nieto. He says, "Hi, Felix, nice talk. You also see an effect of MSC1 mutant in G2M when sisters are not yet separated, not only in telophase. Does this fit with your model?"
Yeah, I mean, I don't think we have looked at this mutant in G2M, but probably it's going to have this sort of septum and compartmentalization. I presume it's going to be in G1, in G2 as well. And it's true that there is, although we couldn't pick up this protein in the G2M proteomic assays, but if you remember, we did in parallel like a late mitotic arrest and a G2M arrest here. And then we compared, and this is the one that is enriched just in G2M, and again by Western blot it's sort of enriched, mostly in late telophase, not in G2M. It's true that if you go to the mutant, you compare, there's an effect in G2M. So there's a drop. It's a significant drop from a statistical point of view. Like it's just a measurement of clonogenic survival of 3 independent experiments. Okay. But it looks like it's aggravated in late mitosis. And there's a decay, there's a decrease in the amount of colony forming units that you get after in G2M as well. So it's likely that there's a role in G2M as well.
Different. Okay.
I don't know if that answers the question. I don't. I hope.
I hope that I think that he has seen this difference, and the question is, does it fit with your model? And probably is what you are saying. Probably.
Yeah, I mean, it's true. Yeah, it's true. I mean, it's true that in the mod, that's in G2M cells are aligned. I mean, sister chromatids are aligned. If we go to this system, so for instance, this is a system, the match switching type system, for those who don't know it very well. So, for instance, in this system, you don't have to go for your sister.
Yeah,
it's an easy way to measure efficiency of homologous recombination. But in this system, in late mitosis, you can just recombine with intramolecularly with a different part of your the same chromosome. But still, you have to bring this arm close to this other arm.
Yeah.
And when you got like 2 sisters aligned to each other in G2M, then you, in theory, you have you should have them very close to each other. So, in that sense, this compartmentalization that the mutant gives you should be less influential, should be less important, because it's very unlikely that you are going to trap. You're not going to trap. Maybe in this case, maybe you could trap in some instances part of this arm in a different compartment. Not clear if the septum is not complete, but for aligned chromatids, definitely. I mean, I presume it's not as easy. I mean, it should be, this compartmentalization issue should have like little effects.
Yeah. So,
and maybe that's the reason that the G2M is not that important.
I think that Maria has another question now about the first part.
Yes, yeah, we are sorry that we went back and forth. So from the other part, what you said that, so if I got it right, you see this phenotype with the, I don't know if it's called a phenotype, but when you see the vacuole and the rDNA condensed surrounding it, in situations in which you blocked mitosis, is that correct? But do you think intermediate exists in normal cycling cells, and you are just and it's transient or not? Not at all. It's just when you
something that you don't find. I mean, in normal cells, In normal cells, sometimes, sorry, because I have to go back.
Yeah, that's what
Probably
I'm saying.
it's easier if I saw the presentation and then try to find the right slide. But so in normal cells, sometimes what you get is something like this. So you can have like a extrusion, like sort of like little extrusion of your RDNA, but you cannot reach the complete toroid or toroidal nucleus that you find. So not even in G2.
Yeah.
And if you treat cells with nocadosol, cells need to get to G2M for this to appear. I mean, it's not because you just add nocadosol and suddenly your nucleus is going to become toroidal in space. No, you still need to get to G2M.
I understand. I meant that it maybe this happen. It's just that when you block them when you are in G2M, then you see this enrichment and they continue growing. But I thought that maybe this thyroid, I don't know how you call it, this enlarged nuclei.
Yes.
My question was if it does have a function for the RDNA, and you just happen to enrich it when you block the cells in G2M, but normally cells growing through G2M, they form a kind of primordia, but then go back or something like that. And if you thought that there has a biological function, let's say in normal mitotic, passing by mitosis cells.
Probably, probably not. I mean, I don't think it's a very remarkable phenotype, standing phenotype. I don't know whether these cells actually, so it's something that maybe you see when you block cells in G2M with nocadosol. Having said this, again, I just wanna.
Because you think there's a couple, this couple, I don't know how to say that, the nuclear, the growth of the nuclear envelope for the other events happening there.
What I think, or we think, is like you have a nuclear envelope which is growing because it needs to grow, because it needs to get elongated in this close mitosis in phase.
Yeah.
You don't stop that growth if you are cells in G2M. This is not only mine, it's just, this was already demonstrated by FIISC lab a few years ago. And then you continue this growth of the nuclear envelope, becomes larger, and in part because you have to keep this volume, what I said about the nucleus to cytoplasm ratio, the way that you do that is, instead of growing your nucleus as a sphere that becomes bigger and bigger, what you do is like sort of you got like wrinkles of your nuclear membrane. So in that way, it's so you continue with this growth, but the nuclear volume doesn't get bigger. And having said this, I mean, well, there's two things which I consider to be important in this. Again, even if this is just sort of a phenotype as a consequence of the G2, this block, this must have a consequences for the cell, which could be exploited in this, maybe this therapy, for instance. And this is something that we are working on at present, is you have this nucleus. What happens to this nucleus if we now allow cells to go through anaphase? And this is something we are exploring, because if the genomic stability during this aberrant segregation, because now it has to segregate in conditions which are not normal.
Yeah.
So if it leads to sort of genomic instability, a loss of viability, etc., again, it could be like maybe somehow exploited in antifungal therapies or anti-close mitosis therapies.
Yes.
And the second thing, I mean, this is something interesting, is like there are many studies now in yeast using this HUNSC, probably you know, high conformation approaches about gene regulation, and many of them are performed in cells which are arrested in G12. And maybe you look at, oh, well, you knew this, you change this pattern of transcription, or maybe you add nocadosol, or you arrest your cells in G12, and you got a shift in your transcription program. But just remember, your nucleus is gonna change its shape dramatically. And this has a consequence, immediate consequence, on how your chromatin is gonna be distributed within the
Indeed, I wanted to ask you if in this I don't know how to handle you call it,
loop, handle, horseshoe.
If there was any transcription going on there. I know it's mitosis; they are condensed, but sometimes you have transcription, and specifically for the ribosomal RNA, if you have any transcription there, did you measure that?
As far as we know, there should be transcription going on. Because it's known that cells which are arrested in nocadosol, the transcription of the RNA continues. It's only after you enter anaphase, you activate CDC14, which is going to switch off transcription to allow the latest condensation of the ribosomal DNA, which is needed for proper segregation. So in principle, during G12, there should be transcription going on. And as far as I know, I think has been demonstrated by many other labs, including probably Andres. I don't know if Andres is here. Probably he knows more, but I'm pretty sure it is transcribed in G12, in nocadosol arrested cells.
But from this handle, you think, can you do FIISC in this hand?
FIISC for transcription.
Yeah. Of the RNA units.
Yeah, well, I mean, it's normally for yeast, the way the transcription, active transcription has been measured is by a quantitative PCR so when you got the RNA transcription, you got like stable transcript and unstable intermediates. If you go for the unstable intermediates, if you still have them there, is that because you got ongoing transcription. Probably there are other ways to do it. By FIISC, I suppose it can be possible. As far as I know, I haven't seen it. But the only way I've seen it so far, if I remember well, is just by this qPCR strategy. It's true that by qPCR, you've got a population approach. So if you want a single cell approach, maybe, I don't know, a couple PCR with this microscopy. Not really familiar with that. I know that it does exist, I think. It exists, but I don't know how to do it.
Okay, so thank you very much to both of you, and thank you also for the audience. We need to close the session and this really interesting discussion. Thank you, Maria, for organizing this, and thank you all the attendants for the discussion, and we hope to see you in the next webinar. And thank you, Félix. Bye.
Thank you. Thank you all.
Vale, estamos ahora